On behalf of the Association for Vascular Access and the I Save That webinar series, I'd like to welcome you to tonight's webinar, Biofilms, a Sticky Problem. I'm Blake Hotchkiss in the AVA Education Department. Joining with me today is Judy Thompson, Director of Clinical Education, and Dr. Lori Burrows, our speaker for tonight's presentation. So Professor Lori Burrows is a microbiologist and international expert on the structure, function, and regulation of type 4 pili. I'm probably saying that incorrectly, but ubiquitous bacterial virulence factors used for adherence, DNA uptake, biofilm formation, and twitching motility. Her group also studies biofilm formation, focusing on stimulation of biofilm development by sub-inhibitory antibiotic concentrations and exploitation of the stimulation phenotype to find new antimicrobials for multi-drug resistant gram-negative bacteria. Burrows' research is funded by the Canadian Institute of Health Research, the Natural Sciences and Engineering Research Council of Canada, the Canadian Glycomics Network, the Ontario Research Fund, and industrial support. She has published over 120 peer-reviewed papers, reviews, and book chapters. She is the Associate Director, Partnerships and Outreach of McMaster University's Michael G. DeGroote Institute for Infectious Diseases Research, a Fellow of the American Academy of Microbiology, and serves on the editorial boards of the Journal of Bacteriology through the ASM, the Journal of Biochemistry, and ACS Infectious Diseases. And with that, Lori, I'll let you take it from here.
Thank you, Blake.
You're welcome.
Thank you for painfully reading that whole bio. I appreciate it. And I also appreciate the opportunity to show you a little bit about what we're doing with biofilms today. So biofilms, a sticky problem. So you guys work on vascular access, and this is a big problem in the vascular access field. So hopefully I can make this relevant to what you work on and work with. I'm going to start off with a disclosure, if I can get my slides to work. There we go. So I'm going to talk about some of the work that we've done in my lab, and the McMaster University has filed a patent on this work, but other than that, I have nothing to disclose. So today's learning objective, since this is a CE event, I'm hoping what you will get out of this presentation is an understanding of what biofilms are and how they form. I want you to learn how biofilms tolerate antimicrobial treatment, but also enable the development of further resistance. And I want you to understand the physical, chemical, and biological approaches that have been tried to prevent and treat biofilms. And I can tell you right now that there are not a lot of very successful treatment modalities out there for biofilms. It's a sticky problem. So I like to start off by just pointing out that biofilms are found on most surfaces. You could actually say moist surfaces in this case. Bacteria like to live attached to surfaces. So in a stream like this, you would have very few bacteria actually free floating in the water. Most of them would be attached to the rocks, and these kind of scum lines at the water line is a heavy biofilm, usually of bacteria that are then being attacked by larger organisms. So if you have a dog or a cat in your house, you'll know that their water bowl gets a slimy ring inside that's really hard to rinse off. You have to actually scrub it off. That is also a biofilm. So biofilms are really sticky communities of bacteria that are found on moist surfaces. They're also found in humans. So this is an older diagram now from a review on biofilms, but I really like the presentation of it. And you can see here that the large majority of human biofilm-related infections are related to medical devices, and that's why this is relevant to vascular access. So, the most common human biofilm infection is actually the one on your teeth right now. So if you lick your front teeth, you'll feel that biofilm growing there right now. And you'll know how difficult that biofilm is to get rid of. You have to use your biofilm removal tool a couple times a day to actually scrub that thing off because it's really sticky and hard to remove. There's a number of medical devices shown on this slide, and we have everything from endotracheal tubes, which are obviously a huge issue right now, to stents, to peritoneal dialysis catheters. And you can imagine that treating a biofilm on some of these devices is a little bit easier than others. So something like an IV line, if it gets contaminated with a biofilm, you can remove it. But if it's something like an implanted joint, it's very difficult to manage a biofilm infection on something that's totally implanted. So, it's better to try to manage, to prevent those biofilm infections rather than to try to manage them. So one of the unique things about the biofilm mode of growth is that bacteria produce what is called a matrix. So this is a coating of, it's a combination of DNA, polysaccharides, proteins, and lipids, and they produce this themselves. So I have two images here. The one on the left is a scanning electron micrograph of Listeria monocytogenes. So this is a food pathogen growing on a stainless steel surface. And you can see it makes these big clumps of bacteria. So using electron microscopy is not the best way to image biofilms, because in order to see the bacteria, you have to dehydrate the sample, and you actually dehydrate the matrix when you do that, because it's normally very hydrated. So on the right side, we have a confocal scanning micrograph of a biofilm, and now you can see this is a hydrated sample. So you can look at the bacteria live while they're still hydrated, and that you have bacterial cells, which are the bright things, embedded in this big slimy layer of matrix. So bacteria produce this themselves, and it really is kind of like the actual matrix, because in this movie, these guys had a whole separate world that was just theirs that not everybody could access, and that's what the bacteria are doing. So I'm going to tell you a bit more later about why they're making this matrix, but that is a fundamental aspect of biofilm lifestyle. So let's talk briefly about the biofilm life cycle. So for a long time, microbiologists studied bacteria in a test tube, sort of free swimming, or what we call planktonic cells, because they're like plankton in the ocean, free swimming, free floating. And we studied them in a test tube because it was easy to grow lots of bacteria to study. But now that we know bacteria actually attach to a surface, we have to think more about how to grow the bacteria in order to replicate how they would normally grow. So the standard sort of picture of a biofilm life cycle is that planktonic cells come into contact with a surface. So they can either actively swim towards a surface using a flagellum or some other motility mode, or they can just randomly float around until they bump into a surface, and then they can attach. So once they attach, they actually undergo genetic changes, so gene expression changes in response to surface contact. And this is a really hot area of research in the biofilm field, because we're trying to understand, what do the bacteria sense when they come into contact with a surface? Is it some kind of shear force? Is it some kind of mechanical signal that causes them to recognize that they're attached to a surface and then start to produce this matrix that I was talking about? So the next step, once they attach, is that they form what are called microcolonies, so little clusters of bacteria, and they start to produce this matrix that starts to surround the colony. And eventually this will get bigger and bigger and bigger until you have a macrocolony. So this can be quite thick. It can be several micrometers, up to hundreds of micrometers thick, depending on how long you leave this. And then to complete the cycle, the bacteria will either break the matrix down themselves, and that's important for what I'm going to talk about a little bit later. So they can make enzymes that will break this matrix open and allow them to escape. Or sometimes the biofilm just gets so big and chunks of it will just break off. So you can imagine, inside a urinary catheter, for example, or an artificial heart valve, if you get a vegetation on a heart valve and it gets too big, the beating of the heart will actually cause chunks of it to break off, and these will float away and start the cycle again somewhere else. So let's talk briefly about why bacteria and fungi, because yeast will also make biofilms. Why do they make a biofilm in the first place? So there are a number of reasons. One of them is that nutrients concentrate at surfaces. So, they're usually moving up a gradient of nutrients, and so they're attracted to the place where there is a higher concentration of nutrients. And if you put, a rock in a stream, as I mentioned earlier, nutrients will tend to accumulate on the surface of that rock, and that will attract the bacteria, and they want to attach there so they can get those nutrients. Growing as a biofilm or as a community promotes communication between the cells. So bacteria don't have eyes or ears, they communicate chemically. And so you can imagine it's hard if you're far apart to communicate chemically because the signals that you're making have to diffuse. So if you're close together, it's a lot easier to communicate with each other. This type of communication, which is sometimes referred to as quorum sensing, can cause the activation of group-specific behavior. So sometimes bacteria will not do certain things or make certain products unless there are enough of them to make a difference. And so this is a way of them actually measuring the density of their own population. So by making these signals, when there's a lot of bacteria, you have a lot of signal that accumulates, and that will lead to the activation of group-specific behaviors. Growing as a community like this allows the easy transfer of DNA fragments between cells, and that is a significant problem for the transfer of antibiotic resistance and even cell contents. So some bacteria can actually fuse their cells to one another and transfer cell contents. But again, if you're growing as a free swimming cell, you can't do that because you actually need to be in close proximity with another cell and for long enough that this can happen. One of the other aspects that's interesting about growing as a community like this is that the bacteria can share what we call public goods. So these, for example, could be enzymes that break down larger proteins to give you amino acids that you can then take up. So often these are things that would benefit the same species, but oftentimes bacteria and fungi are growing in communities with other species. And so sometimes this is good because, species A can break down a large molecule into smaller things, and then species B can take those smaller things and break them down further. And that is useful, for example, in things like wastewater treatment. But it also can lead to cheating, because you can imagine if you have a group of bacteria that need to make an enzyme to break down a food source, and you have mutants that accumulate that don't make that enzyme, they actually get the benefit of their neighbors' enzymes, but they don't actually make the enzyme, so they don't have that metabolic cost. So there is a whole field or subfield of microbiology called sociomicrobiology, where people study how bacteria restrict cheaters from prospering, because we don't like cheaters. And lastly, growing as a biofilm will protect the bacteria from phage, which are small viruses that prey on bacteria, predators such as amoeba, which eat bacteria, immune cells, desiccation, which means drying out, and from antibiotics and disinfectants. And that's one of the important aspects that we study. So before I talk any more about antibiotics and disinfectants, I just want to give you some quick refreshers on definitions. So when I say MIC, I mean minimal inhibitory concentration. And this is what we talk about when we have a concentration of antibiotics that reduces the growth of a bacterium to the point where we can't actually see growth in a tube. So it's a little bit of a subjective measure, because it has to do with the turbidity of the culture and what you can actually see with your eye. MBC is the minimal bactericidal concentration. So this is the minimal concentration of a drug that will actually kill the bacteria, so that if you expose them to this concentration and then tried to regrow them, they would not grow. And lastly, the minimum biofilm eradication concentration, or MBEC, is an important measure for biofilms, because biofilms themselves are much harder to kill with antibiotics than free-floating cells. So before I go on and tell you why they're so hard to kill, we're going to have a quick poll. So I'm hoping that Blake is going to run the poll here.
You got it. I got three
Okay.
separate ones for you, one for each of these questions. So they're all, you know. Let's launch the poll. Are you aware of biofilm and its association with infections?
Awesome. I love it.
Great.
We should have said, are you scared of biofilm and its association with infections?
True, true, very true. All right, let's wrap it up. So 90% said yes, 10% said no. Let's go to number 2. So do antibiotics penetrate biofilms? Yes or no? All right, so we had about 1/3 said yes and 2/3 said no. And, you know, I'm sure there would be a different response if there was a maybe. So. Let's see here, poll number 3. So the third question on the slide. Can biofilm on a central venous access device cause disseminated infections? Yes or no?
Excellent. That is true. I did sort of give that one away though by telling you about heart valves and vegetations.
All right.
All right. Excellent.
97%.
So I'm very happy to hear that people know what biofilm is, but there seems to be a bit of a mixed reaction when it comes to whether antibiotics can penetrate this matrix that I talked about.
Great.
All right. So if you close that poll, I will go on to my next slide.
You should be all set now.
Okay, I still see the poll. Maybe I can close. That's just me. All right, onwards. The biofilm mode of growth provides protection against antibiotics and disinfectants. So when we have bacteria in an antibiotic or in a planktonic state, they are antibiotic susceptible, and this is the state which is used to do MIC testing. So when you submit a sample to the lab and they are testing to tell you which antibiotic the organism is susceptible to, they will grow it planktonically. And that is actually, a bizarre thing that we do because, as I told you, most bacteria grow as biofilms, even in humans, right? So approximately 80% of human infections are biofilm associated. So it's weird that we still do this, and people are trying to change that, but it's difficult to change established practice. So what will happen is, as part of these genetic expression changes that are happening as microcolony formation is going on, those changes lead also to what we call antibiotic tolerance. So I want to make a distinction here between tolerance of antibiotics and resistance to antibiotics, because resistance is what we think about when we have, for example, MRSA or MRSA, methicillin-resistant Staph aureus. That means that every single member of that population is resistant to methicillin. And that is a genetically encoded type of resistance. This type of resistance or tolerance is a function of the mode of growth of the bacteria as a biofilm. So if I take this macrocolony and I disperse it, so I break it up, and allow the bacteria to become planktonic again, they will return to an antibiotic susceptible state. Not right away, because the bacteria that come out of this are still somewhat tolerant, but if you grow them out over time, they will return to the state they were in originally, which is susceptible. So this is more a function of the mode of growth. So how does this manifest itself? Well, first of all, and that's why we asked you this question, there is limited penetrance of some antibiotics into biofilms. So not all antibiotics, some antibiotics. So for example, a lot of the matrix, as I mentioned, is DNA and polysaccharides, and these are often charged molecules. And antibiotics that have the opposite charge will have a very hard time diffusing through this type of matrix. So a typical example are aminoglycosides. So aminoglycosides are positively charged. If you have a negatively charged DNA and sugar matrix, they're going to get trapped in that matrix. Other antibiotics like fluoroquinolones, like ciprofloxacin, for example, do not seem to have much trouble diffusing into the matrix. So penetrance does not explain all of the tolerance that we see. One of the interesting things that happens is when you get a big macrocolony like this, or a thick biofilm, you start to see physiological heterogeneity. So that's a very wordy term that just basically means that some bacteria are not the same as other bacteria. So even if this was all one species, you can imagine as a function of where they find themselves in this biofilm, some of them will be growing more rapidly than others. So this guy is sort of on the outside of the biofilm. He's got more access to food and more access to oxygen, whereas a guy in the middle may have less access to food and oxygen and be growing more slowly. And most of the antibiotics that we use target physiological processes that are happening when the bacteria are growing. So DNA replication, making messenger RNA, making proteins. And if you are not growing very rapidly, you are not really doing those things, and that makes you tolerant of the antibiotic. So that's an important aspect. Just the interaction with a surface induces stress responses in the bacteria. And some of those stress responses help repair damage in bacteria that are exposed to antibiotics. So the act of attaching to a surface and the induction of these stress responses will also help the bacteria tolerate low levels of antibiotics. And lastly, there is this unique type of cell that forms in a biofilm called a persister cell. So we're still trying to understand exactly what these cells are, but essentially they are cells that are almost dormant completely and are very tolerant of antibiotics. They're almost like a spore, if you will. And you can kill most of the biofilm if you get the antibiotic concentrations high enough, but you really cannot kill these cells very easily. And once you take the antibiotic away, those persister cells will then regrow. So these things together contribute to this antibiotic tolerance. The other thing that happens in a biofilm, though, is because of this tolerance, you provide an environment that leads to the development of antimicrobial resistance in the traditional sense. So because less drug gets into the biofilm, there is less drug exposure against the cells, and that gives them time to acquire mutations that lead to resistance. These stress responses, some of them are damage repair pathways, and those damage repair pathways are not always super careful. Sometimes they're what we call error prone, and that can also lead to mutations. So the bacteria are more interested in repairing the damage quickly than repairing the damage well in order to survive. So that can lead to mutations that ultimately give you resistance to antibiotics. And as I mentioned earlier, one of the benefits for the bacteria of being in close proximity like this is that they can exchange DNA easily. So they can exchange plasmids and transposons that encode resistance genes. Because they're in close proximity, pretty much if you put one cell into a biofilm that has a plasmid that can be transferred within a short amount of time, all of its neighbors will have acquired it and so on. And pretty soon everybody has this resistance plasmid. And we understand now that you can get resistance exchange, not just between organisms of the same species, but across species as well. So this is one of the main mechanisms by which antimicrobial resistance spreads. So the other weird thing that happens is that subinhibitory concentrations of antibiotics and disinfectants can actually stimulate biofilm formation. So, whereas making a biofilm will help the bacteria resist or tolerate antibiotics. Exposing them to too little antibiotic will actually stimulate biofilm formation. So my lab studies this problem, and we sort of stumbled across it accidentally, like most good serendipitous things in science. We were looking for small molecule inhibitors of biofilm formation that we could combine with antibiotics in order to improve the killing of the antibiotics. So our idea was that we would give the antibiotic at the same time as a biofilm inhibitor so the bacteria would not have time to form a biofilm and resist the antibiotic. And what we found instead were biofilm stimulators. And I don't have a ton of time today to tell you the whole backstory, but essentially what we realized is that these biofilm stimulators were actually sub-inhibitory antibiotics. And that, what the mental picture that I have now is that, a lot of antibiotics are made by soil organisms. And you can imagine, if you're a soil organism making an antibiotic and you're a bacterium growing towards it, you want to start expressing a phenotype that's going to help you survive the antibiotic that this organism is making. So as you're growing towards it, by forming a biofilm, when you detect low concentrations, you can protect yourself. So I'm going to just mention briefly the assay that we use to measure biofilm formation in our lab. And this is called surprisingly, the MBEC assay. So remember I mentioned the minimal biofilm eradication concentration. So this is the MBEC device, or the Calgary biofilm device, because it was invented by a scientist called Howard Seary from the University of Calgary in Canada. And what it is essentially is a special lid with 96 pegs on it that fits into the wells of a 96 well plate. And we really love this assay because it allows us to study, for example, 96 different drugs or 96 different bacteria or concentration gradients of drugs. And you get biofilm formation by the bacteria on the pegs, and then you can remove the lid, wash off any bacteria that are not firmly stuck to the peg, which is, they usually are quite stuck to the peg. And then we basically dunk this lid into a solution of crystal violet, which is a purple stain. It's the first step of the Graham stain for those of you who remember your basic microbiology. And the purple dye gives us an indication of how much biofilm has formed on the peg. We can then take the stained pegs and again, immerse them in a new plate that has either ethanol or acetic acid in it, and that will elute the dye, and then we can measure the dye with a plate reader, and that tells us how much biofilm we have. So it's an indirect measure because it's, there's multiple steps there, but it is very reproducible, and we really like it. So I'm going to just mention briefly some of the initial studies that we did that alerted us to this biofilm stimulation phenotype of antibiotics. So this is an example of a very common antibiotic, ampicillin. And when we do drug screening in a high throughput screening environment, you can't screen every drug at every concentration. So typically we would screen drugs at a concentration of 10 micromolar, which you see at the end of this first graph here. And for ampicillin, that is a pretty high concentration because the MIC for ampicillin, the minimal inhibitory concentration, is down at 0.3 micromolar. I'm going to show you a lot of these graphs, so I should stop and just take a moment to mention that yellow bars are the growth of the bacteria and purple bars are the biofilm. So when we use this lid, well, let me just flip back here for a second. When we use these lids, we also measure the growth of the bacteria in the 96 well plate itself, because we want to know if a bacterium did not form a biofilm on a peg, is that because we just killed it with the drug? Or is it because that particular drug inhibits biofilm formation, although it does not inhibit growth of the bacterium? So there's a distinction there. So in this case, 0 is our control, and that is 1% DMSO, because that is the solution that all of our drugs are resuspended in. So we did this experiment and we killed our bacteria beautifully, but in other experiments, we saw that if we diluted the drug more, we would still get biofilm. So we decided to take this range right here at the lower end of this concentration spectrum and expand it a little more. So this graph over here starts at the highest concentration of 0.63 and goes down. And what we saw, to our surprise, was that when you dilute the drug below the MIC, you don't just get the same amount of biofilm as control, you actually get more biofilm than the control. So the student that was doing these experiments came running into my office and was completely horrified because, of course, she was supposed to be looking for biofilm inhibitors. And she said, I'm making it worse. And I was like, no, this is cool. Let's figure out what's going on here. So if you take the PEG from this 0.04 micromolar ampicillin sample, and look at it with an electron microscope, which I mentioned earlier, you can see that it is absolutely loaded with biofilm compared to the control. So this dark gray material here is the actual plastic PEG in the background. And you can see normally you would have little clusters of bacteria like the image I showed you earlier. And here you just have massive amounts of bacteria. So this is a really fascinating phenotype, and it is kind of scary actually, because you can imagine if you give too little antibiotic, you are actually going to make this problem worse. So we need to know that this is happening. So I think of this as almost an anti-antibiotic phenotype. So in other words, if you expose the bacteria to a concentration of antibiotics that doesn't kill them, but starts to cause damage, you will trigger this biofilm stimulation response, because as we know now, biofilms will protect the bacteria. They'll help them to tolerate higher concentrations of antibiotics. So this is almost like a quick response they can do in order to protect the population while they manifest more elaborate resistance mechanisms. So the one I just showed you was Listeria monocytogenes, the foodborne pathogen, but this is not something that is limited to Listeria. So this is an image from a paper from another group, Kaplan et al., with MRSA. So MRSA is methicillin-resistant Staph aureus, and they looked at two different strains here. These are very common strains that are circulating in the US. And they're looking here just at the biofilm formation in a colorimetric way. So if you don't expose these bacteria to the drug, they form very little biofilm. So this is kind of paradoxical, right? So we think of these bacteria as good biofilm formers, but in the absence of drug, they don't want to waste the energy to make a biofilm. As soon as you start to increase the concentration of methicillin, they immediately make tons of biofilm, just like I showed you for Listeria. Eventually, though, you will get to a high enough concentration that you kill the bacteria, and then, of course, you don't get any biofilm from dead bacteria. So this is a common phenomenon, and this is methicillin. I showed you ampicillin, but this is not just limited to that class of drugs, so the beta-lactam class of antibiotics. So this is data for the organism that my lab works on, Pseudomonas aeruginosa. This is a gram-negative opportunistic pathogen. I'm sure if you guys are working in hospitals, you will be very familiar with this bug. It's a very drug-resistant organism and very hard to treat, and it's on the World Health Organization's list of top bad bugs. So when we expose Pseudomonas to subinhibitory concentrations of ciprofloxacin, which is a fluoroquinolone, cefixime, which is a cephalosporin, or erythromycin, which is a macrolide, in all cases, we see the same sort of, as we approach the MIC, so as the growth starts to drop off, biofilm spikes before we get to the MIC. So it's the bacteria going, oh my God, I'm gonna die, and then they die. But in the meantime, they are trying to protect themselves before they hit that concentration. And we see this reproducibly for almost every single drug we've looked at. We even see this for things like soap. So this is an image from a study that we did for a company that was developing a surgical scrub. And this is just a very mild Castile soap type of product. And if you use it at 100%, it's very antimicrobial. But if you dilute it, you'll see you eventually will get down to a subinhibitory concentration, and that will cause biofilm to spike again. And this makes sense with some of the literature. If you look in the literature, you'll see that sometimes if hospitals are cheap and they dilute their hand sanitizer or their soap too much, you will get bacterial growth in it because you're causing this phenomenon to happen. So we've even seen biofilm stimulation with compounds that are supposedly not even aimed at bacteria at all. So these are two examples. So loperamide is Imodium, which is an anti-diarrheal drug that causes biofilm stimulation in bacteria. Flufenazine, which is an antipsychotic drug. Again, we see the stimulation of biofilm. So we're not killing the bacteria with these drugs, probably because we're not at a high enough concentration, but they definitely are sensing and responding to those drugs. So there are several other examples I've listed at the bottom here, but we have a collection of compounds at McMaster that we screen frequently, and this is approximately 4,000 FDA approved but off-patent drugs that are meant for doing repurposing screens. And so that's where these came from. So, we have to be aware that even though we're giving people drugs that are aimed at treating the people, we can sometimes be accidentally treating the bacteria, because at the time when these drugs were developed, people checked for off-target effects on the humans, but not necessarily off-target effects on the human microbiome, because a lot of these are very old drugs, and we didn't have a good appreciation at the time that there was a human microbiome. So we're not the only people that have seen this phenomenon. This is a paper that came out a couple of years ago from a group in Europe, and they essentially tested the same collection of drugs on about 40 different bacteria isolated from the human gut microbiome. And they saw similar things, that a lot of these bacteria responded to drugs that had nothing to do with antibiotics. So, this is something to keep in mind. So we've been trying to figure out exactly how sub-MIC antibiotics stimulate biofilm formation. So it doesn't seem to be aimed, because so many different classes of antibiotics with different targets and different mechanisms of action can cause this phenotype, it doesn't seem to be related to any specific inhibition of any specific process. It seems to be a more generic response that is kind of a way of bacteria to panic and make a biofilm so they can protect themselves from this toxin that's coming at them. So this is an image from a review article that we published a couple of years ago now, just sort of laying out some of our hypotheses about what might be going on here. So the first one that we proposed was that sub-MIC antibiotics can cause damage, and that damage will trigger the induction of prophages. So prophages are bacteriophages or viruses that have integrated themselves into the genome of their host. And they basically ride along as long as the bacteria is healthy and get replicated as part of the bacterial genome. And this is a very lazy way for them to propagate themselves. However, they have mechanisms to sense when their host is damaged, right? So if the ship starts to sink, these guys are out of there. And so they will come out of the genome and they will lyse the cells and try to find a new host. And so this, it's been shown that antibiotics can induce prophages. And so when they do this, you can imagine that the debris that they release when they lyse their host can seed biofilm formation. The second hypothesis is that, remember I mentioned that the bacterial population is physiologically heterogeneous, and some cells, because they're growing faster than others, will be more susceptible than others. And so we had the idea that maybe sub-MIC antibiotics kill a subset of bacteria, and the debris from those guys will seed biofilm formation. And then lastly, we know that sub-MIC antibiotics trigger stress pathways. And it's possible that there's a link between stress pathways and biofilm specific promoters that lead to matrix production. So these hypotheses are things that we're working on. We still don't know which one is true. We're sort of leaning towards number 3, but these are not mutually exclusive either. But while we're trying to figure out what's going on, we wondered if we could actually exploit this phenotype to detect sub-inhibitory antimicrobials. So in other words, you know, can the ability of bacteria to make a biofilm even before you get to a concentration of antibiotic that will kill them be used as sort of a canary in a coal mine to alert us to the fact that a drug might actually have antimicrobial activity, but just be at too low of a concentration. So as I mentioned earlier, we normally screen, when we do high throughput drug screening, we normally screen at a single concentration, which is typically 10 micromolar. So this is erythromycin and exposed against Pseudomonas. And if we screened erythromycin at 10 micromolar and looked just at growth, we would say this is not an antibiotic, because I don't see any difference in the growth between 0, which has no drug, and 10. However, if I look at the biofilm formation that's made by Pseudomonas at difference between 0 and 10, I can see now at 10, which is well before the MIC of erythromycin for Pseudomonas, which is, you know, way up here at like 160 to 320. Already I can see this, oh my God, I'm gonna die phenotype. And so that will alert us to the fact that, hey, this is actually potentially an antibiotic, but it's just at too low of a concentration to kill. So we've been using this phenotype to look for new drug activities in this previously approved drug library that I mentioned. So I'm just going to give you a quick story about one of the drugs that we found. So this is a data set, or a sub data set from that screen. And these are just 2 replicates of screening looking at biofilm formation. And we're, you know, most drugs will not affect biofilm formation. So you're down here at 1. So a ratio of 1 means nothing's happened. But we're really interested in things out here that affect biofilm formation. So some of these obviously are antibiotics, because a previously approved drug library will have antibiotics in it. But one of the compounds that really stood out to me when we looked at this data set was this drug thiostrepton. So this is a really old antibiotic, but it is not known to have any activity whatsoever against gram-negative bacteria. And we were screening this against Pseudomonas. So I was really surprised by this. And the reason that I was really surprised is because this drug is huge. It's huge. So it should not be able to get into Pseudomonas. Pseudomonas is very good at excluding large molecules like this from getting into the cell. So the fact that we saw this, you know, very typical biofilm stimulation curve for this drug surprised me. So we went on to study whether this drug was actually getting into Pseudomonas, and it was, and whether it was killing them, and it was, and how it could do that. And long story short, we figured out that this drug is a mimic of the bacteria's iron uptake molecule pyoverdin. So Pseudomonas is well known for being green. This is really pretty green color on plates. And it's green because it makes this molecule pyoverdin, which is a green fluorescent compound. And it makes this compound because it's trying to get iron. And this sticks to iron, and the bacteria take it up into the cell, and that's how they acquire iron. Well, it seems that this molecule here has evolved to hijack the receptor for pyoverdin. And so we figured that out. This is just a graph showing different mutants lacking different iron uptake receptors. And it wasn't until we got rid of both pyoverdin receptors, because Pseudomonas has 2, that we saw resistance to this drug. Everything else, all the other mutants were killed. But once you take away basically the 2 doors that this drug uses to get into the cell, then the bacteria survive. So this was a really cool story. And we've gone on to study this a little further. We showed that because this drug is taken up under conditions of low iron, we combined the drug with FDA-approved iron chelators. So these are molecules used to treat people that have iron overload disease. So deferiprone is one, and deferocerox is the other. And these are called checkerboard assays, where you have a dilution of one drug in one direction and the chelator in the other direction. And you can see that on its own, this drug isn't great at killing bacteria. But if you combine it with the chelators, then it becomes much more powerful and you get killing of the bacteria. So Deferocerox works really well. So on its own, Deferocerox doesn't kill Pseudomonas, and on its own, Thiostrepton does not kill Pseudomonas, but combined, they are wicked. I love it. The other thing we tested was just serum, mouse serum and human serum, because human and mouse serum is inherently restricted in iron. So you don't even need the chelator in that case, right? As soon as you add the drug in the presence of serum, it kills the bacteria. So we have, this is just one example of many things that we have identified using that biofilm stimulation phenotype to alert us to the fact that they have activity. Okay, so in the last part of my talk, I just want to talk briefly about how you can manage biofilm infections. So they are really hard to manage. I'm just going to put that out there at the beginning. So, you know, I teach a class on biofilms every year, and every year I ask the students to come up with creative ways to get rid of biofilms, and they always come back to antibiotics and disinfectants. So, you know, that just shows you how challenging this problem is. So I sort of broke down the treatments or the prevention strategies into physical, chemical, and biological. So I'm going to go through those, but I want to emphasize that prevention of biofilm formation is key because treatment is difficult. So, you know, at a bare minimum, you need to have exceptional sterile technique when you are using medical devices, because you do not want to contaminate them with bacteria. It's unavoidable, of course, but, you know, do your best. So let's talk about physical ways to reduce biofilm formation. So there are studies out there showing that low electrical fields can reduce biofilm formation. This has been tested in urinary catheters, but, you know, personally, I would not want an electric urinary catheter inserted, but they've used this kind of strategy in industry. So, for example, in dairy factories where they're managing milk products and they have lots of pipes where they have to move milk from A to B. Milk has a lot of microorganisms in it. And so by wrapping electrical fields around the pipes, they can actually reduce the biofilm that forms on the inside of the pipes. Ultrasound is a really common way to deal with biofilms. So, you know, we go to the dentist and they use ultrasound to clean our teeth. We use ultrasound to clean jewelry. In doing that, we are removing biofilms. So this is actually a useful technique. And ultrasound is also helpful in helping antibiotics penetrate biofilms. So I would say that is a useful strategy. One of the newer methods that people have been thinking of is to change the topology of a medical device in order to help reduce biofilm formation. So I want to show you a little video of what I mean by that. And that means, you know, changing the way the surface is shaped without actually adding anything to it, right? So you don't have to add drugs or anything to the surface. You just change the way the surface looks to the bacteria, and you try to manipulate the bacterium's behavior. So you remember I told you, you know, the first thing they have to do in order to make a biofilm is attach to the surface in the first place. So we're messing with their ability to attach. So let me show you this video. This is a video that comes from the lab of Cynthia Whitchurch, who used to be in Australia and is now at the Quadram Institute. And they came up with this really clever way of causing Pseudomonas to be confused about which direction it was moving in. So this is twitching motility, which Blake mentioned in my introduction. Pseudomonas can crawl across surfaces very rapidly. So you can see the time clock ticking there. And it's spreading super, super fast on a smooth surface. But Cynthia noticed that the bacteria would follow grooves in a surface. So just like, you know, if you go for a hike, you don't just randomly walk into the woods. You follow the trail that other people have made before you. And so bacteria do the exact same thing. And by taking advantage of this property, you could fool the bacteria into following these grooves. So you can imagine if you have a catheter and you have these grooves going around the catheter, instead of spreading up, the bacteria will start to spread and then they'll just go around in circles and follow these grooves. So this is a really cool concept for a drug-free way to reduce bacterial adhesion to a surface, or to at least slow down biofilm formation. Okay, so that's physical methods. How about chemical methods? So chemical is probably the most common way people deal with biofilms now. And I can tell you, after 21 years in this field and trying to make various surface coatings that would resist biofilm, is very difficult to do. And that is because no matter what you put on a surface, you will eventually get coverage of that thing with what is called a conditioning film. So if you put a clean medical device surface into a person, you are going to get all kinds of human molecules deposited on that surface, and bacteria will then adhere to those. Many people have tried to make surfaces that elute things like antibiotics. That is not a good idea because, as I mentioned, that can contribute to resistance. So those approaches are sort of frowned on now. Another common chemical coating is silver. It has some advantages, but it also has some disadvantages because silver can also be toxic. And we are now starting to see bacteria that are resistant to silver. They have modified efflux pumps for other metals like copper and use those to resist silver. So the more we use silver, the more we're going to select for that. One of the common things, especially in the case of vascular access, that you're going to see is lock solutions. And I think these are actually a good way to prevent biofilm. So I'm just going to give you a quick example here of 4 different common pathogens. And this is showing biofilm eradication by a lock solution that contains 4% tetrasodium EDTA. So staph epi, very, very common skin organism and a big problem on lines. This thing can be eradicated using this solution in 3 hours. Methicillin-resistant Staph aureus, that's a bit more of a tough guy to kill, but again, with slightly longer exposures, you can get rid of these biofilms on surfaces. These experiments, by the way, were done on the same type of pegs that I showed you earlier. Pseudomonas and Klebsiella are gram-negatives, and they are actually more susceptible to this. So EDTA chelates divalent cations, and these guys have an outer membrane that is held together by calcium and magnesium. And so when you start to chelate calcium and magnesium away from them, you make them very porous and they have a hard time surviving. So this type of catheter lock solution is great because it will actually get rid of biofilm that's pre-existing as well as preventing biofilm formation. We're going to stop here and have another poll.
Woohoo! Let's see here, poll number 4. Is your current CVAD locking solution anti-biofilm? I did add a I'm not sure one to this. So let's see what people say.
Interesting. So I'm guessing people are using things like heparin or citrate. Those things do help a little, but they're not quite as good as this tetrasodium product.
Let's share the results. So 58% said no, 8% said yes, and 35% said I'm not sure.
You got one more for me?
One more poll.
Right on.
Does your current locking solution eradicate biofilm? Also an I'm not sure response to this one.
Definitely a no there. All right. You guys need something better, but hopefully you can understand better why that's such a problem. Excellent. All right. So that's mostly a no.
Yeah. Big old 70% on no, and 28% says I'm not sure.
Excellent. All right. I'm just watching the time here. I'm almost done. So the last strategy I want to mention is biological. And by this, I mean, basically not chemical and physical. We have probiotics. So probiotics are a pretty new approach to inhibiting biofilms. And I would say it's probably most common in the dental biofilm industry. So there are some probiotic strains of lactobacilli that you can use to displace or inhibit the growth of streptococci in your mouth that cause dental caries. So Streptococcus mutans in particular. So this is something that, it's basically a bacteria on bacteria warfare type of scenario. And I don't see how that would be too useful for vascular access. So I'm just going to skip over that. Bacteriophages are becoming sexy again. So they sort of fell out of use when antibiotics came along because they're very specific for certain bacteria, and antibiotics are a little more broad spectrum. So people found antibiotics easier to use. But since we are now in the era of superbugs and multi-drug resistant bacteria, people are turning back to phages. And so there's a lot of interest in looking for phages that can actually chew their way into a biofilm, because you can imagine phages and bacteria have evolved together for a very long time. So the phages have figured out how to do this. And so we're trying to understand, can we find phages that will chew into a biofilm and kill the bacteria? And also, how do they do that? Because maybe we can take advantage of the enzymes that they make that will chew their way in. So the other types of enzymes that I want to mention briefly here are enzymes that the bacteria themselves make in order to escape from a biofilm. So remember I showed you the biofilm life cycle at the beginning, and the dispersal event sometimes is triggered by the bacteria themselves. What if we could take advantage of those enzymes that the bacteria make themselves? If we add them to the matrix, maybe we can actually dissolve it. So one of my colleagues, Lynn Howell, has done exactly this. They took the enzyme that the bacteria make to degrade their own matrix and made large batches of it. And when they add it to the biofilm, you can see the biofilm dissolves, it falls apart. And so they have been trying to combine these matrix enzymes with antibiotics, and also to attach these matrix degrading enzymes directly to the surface of medical devices to try to essentially, as soon as the bacteria attach and try to make a matrix, the enzyme is already there and it will chew up that matrix before the bacteria can make it. So I think it's a really cool approach. It's a little more expensive to make proteins. So, it's a little more elaborate of an approach, but we will take what we can get, especially if we're having issues. Okay, so I am at my summary slide, and I just want to leave you with these take-home messages. So biofilms are the most common mode of growth for bacteria, and that is because they protect the population from many stresses, including, in our case, antibiotics. They are very difficult to remove once formed. They're very sticky, and it's much better to prevent them than to try to get rid of them, right? So anyone, if you've ever brushed your teeth, which I'm sure you have, you'll know that it's the next day the bacteria are back. You cannot get rid of all the bacteria. They seed disseminated infections because you have these either single cells or chunks of biofilm that will break off and move to new locations, because that's how they progress. And they will contribute to the development of superbugs. So prevention is key. I just want to briefly thank the students in my lab who do all the work, and I just get to talk about it, and the funders that fund our work. And I am very grateful for your attention, and I will be happy to answer any questions.
Awesome.
Great presentation. Thank you so much.
Sorry, I'm a few minutes over, but that's because Blake talked for a really long time at the beginning.
I know, he tends to do that. He tends to do that. So we're going to start with the first one that came in. How fast does biofilm grow inside an IV catheter?
It's a good question. I'm going to give you a wishy-washy answer. So it depends on the strain, the organism, the fluid that's going through it, and what the initial load was that got in there. So, if you really are sloppy and you get a lot of bacteria in there, it's going to grow pretty fast.
What's pretty
Um,
fast?
so, it depends on the organism, right? When we grow a culture in the lab, we can get, 10 to the 11 bacteria overnight in a tube. Like, in fact, you could, if you grow bacteria in a tube with media that they like, you'll get a thick biofilm in there after 48 hours that you can squeeze out like toothpaste. It's
Nice.
nasty.
Even better.
Nice.
So I would say faster than you would like. How about that?
Okay, that sounds great.
So, what do you think about using vancomycin in very high concentration to lock an IV catheter to get rid of biofilm?
What do I think? So I don't love the idea of using antibiotics to get rid of biofilms unless that's your only choice. So, we are trying really hard to preserve the antibiotics that we still have because we are losing them to resistance faster than we're making new ones by far, right? So most pharmaceutical companies are out of the antimicrobial game, because why would you make a product that's going to be lost to resistance when you can make Viagra? So you can use high dose vancomycin to do that. It works only against gram-positive bacteria, which is another problem, right? So gram-negative bugs like Pseudomonas. You remember I showed you that crazy Thiostrepton that we're working on? I mean, vancomycin is equally as large and doesn't really get into gram-negative bacteria very easily. So you're limiting yourself by using a drug that will only target 50% of the potential pathogens you're dealing with.
Do you know if biofilm, thank you for that answer, by the way, biofilm is attributed to the majority of implanted ports that are discovered to end up being infected, especially those that are newly implanted?
Mm-hmm. So it's very difficult to get rid of all skin organisms when you're putting something through the skin. So, I'm not surprised that most devices will get contaminated eventually. I mean, that's probably the primary reason for failure, right? Patency, the other, but contamination, biofilm is the major cause. So, things like peritoneal dialysis catheters, they don't have a very long lifespan because of this problem, right? And no matter what people have tried to put on them, it eventually will fail.
So it's an interesting point that you make. So what happens with biofilm in the patients who are able to maintain their vascular access device for a longer period of time? They don't have any complications. They're on long-term antibiotics or long-term therapy, and they don't have any issues. Is biofilm an issue with those folks? Are they?
I would say they probably have some kind of a slow growing biofilm, because I don't think I've ever seen a medical device that's been in place for a while that doesn't have bacteria on it. For example, urinary catheters is a common example in nursing home environments, right? So you can have a urinary catheter in that has bacteria on it, and the patient has bacteriuria, but they don't have any symptoms like fever or things that you would worry about, right? So even though they have bacteria on the catheter and in their bladder, we just leave it alone unless it becomes a problem. So your level of concern has to be related to the types of symptoms that you're seeing, I would say.
So you were talking about the smooth surfaces.
Mm-hmm.
So are groove surfaces,
groovy.
That make it groovy? So they're groovy. Does it make it more difficult to grow biofilm?
So the bacteria is still attached to the surface in a similar way, but they wander around a lot more, right? So that type of approach is good for bacteria that are motile and that are crawling. Other groups have tried to make surfaces that are inherently difficult for bacteria to attach to. So they often model these surfaces on natural surfaces. So shark skin is a common one, right? So sharks are very good at keeping bacteria from sticking to their skin, even though they're immersed in water all the time. So there's a company that makes shark skin-like surfaces. There's another group that are trying to make surfaces that are more like super slippery. So they use the pitcher plant as an example, because pitcher plants trap insects because the insects slide right into the pitcher part of the plant and they can't get out again, right? Because they're so greasy and slippery. So they try to understand the properties of those surfaces. And then a third approach is to make a surface that has very dense spikes sticking up, sort of like a bed of nails. Because you can imagine a bacterium, if this is a surface, a bacterium has to like lie down on the surface, right? And so if the surface is spiky, it can't attach properly. Those type of surfaces don't stay clean very long though, because again, you get a conditioning film. And so the, you know, your nice spiky surface gets all clogged up with guck and then it doesn't work anymore. So
Guck.
good idea, but not a long lasting one.
I know for a lot of folks out there concerned about phlebitis and DVT formation, the spikier surfaces might precipitate that kind of event.
Yeah, exactly. I didn't think of that. Yeah.
Speaking of device material, how well would CHG or chlorhexidine gluconate impregnated catheters work for this purpose?
Again, they do work. We worry again about elution of disinfectants and antimicrobial molecules from these surfaces and what that means for resistance. And I know chlorhexidine in particular in some patient populations has some toxicity issues. So I don't know that that would be my favorite approach.
I know there's some concern over chlorhexidine sensitivity in patients, and
Yes.
that seems to be a common theme nowadays with manufacturers, even just the sponges we put on the outside of the catheter to prevent infection. So,
on the EDTA, what's the minimum dwell time in a catheter for it to be fully effective?
So that paper that I have cited in my presentation has a number of other graphs in it that address that particular question. So I would encourage the person who's asking that question to take a look at that paper. It's in M Systems, or sorry, M Sphere, 2018, Liu et al. So there are dwell time studies in there, as well as studies with, I only showed a few graphs, but they've also looked at the ability of that solution to inhibit the biofilms made by fungi, which are fungi scare me. They're very hard to kill. Sorry, that was a very long answer to a very short question.
EDTA not available in USA yet. How will I get it?
Well,
Good question.
let's see. Talk to Sterile Care.
Yeah.
I think that might be part of the reason why this webinar is going on right now, I would believe it's hopefully coming to the US soon. Your thoughts about EDTA impregnated into catheter surfaces?
Again, probably would work great for the first little while until a conditioning film forms on it. So, you know, in my experience, no one has been able to find the ultimate Teflon of catheters that the bacteria can't stick to, despite a lot of efforts in that direction. So, you know, you can't keep surfaces clean in a body. That's just reality, right? You know, I've even seen people try to force the orientation of human molecules because, you know, bacteria recognize things like fibronectin using specific receptors. And so they try to force the fibronectin to be in the wrong orientation by putting antibodies on the catheter to hold it in the wrong orientation. But, you know, that just seems like it's going to be a $500 catheter that still may not last very long. So, you know, at the end of the day, the technology that you use has to be cheap, right? Because we're using a lot of it and nobody wants to spend any more money on medical devices than they already are.
What do you think about the difference between 2% and 4% EDTA lock solution?
From the data that I've seen, 4% is better than 2%. That's about all I have to say about that. I would say, you know, if you have specific questions about the 4% EDTA solution, you should direct them to sterile care because I don't want to speak on their behalf. They are for sure the experts on 4% EDTA.
Thank you. We recently have some trouble with the Ceniobacter from pigs. What is the best option to lock pigs? Currently, we don't have any solutions.
Yeah, Acinetobacter is a really nasty gram-negative bacterium. It's sort of related to Pseudomonas in that it's, you know, it's actually the World Health Organization's top bad bug for which we need new drugs. So I wish I could tell you what to use. I can tell you that the thiostrepton chelator combinations that we are working on will kill Acinetobacter because it has the same type of pyoverdin receptors that Pseudomonas has. So, you know, those types of drugs may be useful down the line. They're not formulated right now for human use. They're only formulated for animal use. So, you know, if you know a friendly veterinarian and you're desperate, maybe that could work.
Fair enough. I have a good vet. But is it safe to flush EDTA into the central system? You can't aspirate it, so we have to flush it in.
I don't know. I don't know. I think that would probably be a volume dependent question.
Is there any benefit to prophylactic antibiotic blocking?
I would say, how are you going to decide which antibiotic to use, right? Then you'd have to have a crystal ball to know which pathogen you're eventually gonna get a biofilm with.
Right.
So, I mean, staph epi is probably one of the most common ones for lines that are going through the skin, just because it's everywhere. And you know, there are different strains of epi that are more worrisome than others because some of them are better biofilm formers than others. But you know, I don't think a lot of people appreciate this exposure to sub-inhibitory concentrations of antibiotics can actually provoke biofilm formation, right? So to me, that's a bit of a dangerous strategy because if you have an organism that is pretty tolerant of the antibiotic already and then you treat it with a locking solution that has antibiotic, you may actually encourage biofilm formation, which is the opposite of what you're trying to do.
Even if you're operating within your institution's biogreen, right?
Yep.
The antibiogreen.
Yeah, it's hard to predict what you're going to get, right? So, you know, something a little more generic like EDTA to me makes more sense because it has effects against multiple organisms, right? Even, you know, I'm sure if you jacked up the concentration high enough, way beyond 4%, it would also affect humans. But, you know, at the concentration that we're using, it's safe.
Someone was mentioning they had missed the first part of the webinar, and they said, I've heard heparin might have the ability to increase biofilm.
Mm-hmm. Potentially. Again, it's very organism dependent, right? So you have to sort of have a strategy that looks at what the most common organisms are that you're dealing with in your setting, right? Because every hospital has its own flora, right?
If biofilm is present on jewelry, is that a concern for infection prevention strategies?
Interesting question. And I would say potentially, right? So, you know, this is why we are told you're not allowed to wear any jewelry when you have surgery. I had some surgery in the last year and I had to get my wedding rings off, which was a challenge. But yes, any surface that's in continuous contact with your body will have bacteria on it. When I started making my slides, you know, back in the day to teach biofilm lectures to undergrads, my daughter was a teenager and was trying to convince me to let her get a tongue piercing. Terrible idea. Right? You have tons of bacteria in your mouth and now you're putting in a surface that they can adhere to. And it's very close to your vasculature of your brain. So I showed her all kinds of horrible brain abscess photos to try to discourage her from doing this. It worked.
So Here's a question. My understanding is that biofilm immediately starts with any introduction of something into the body, even with sterile technique. So even under the best circumstances, is that true?
So you're going to get a conditioning film immediately, which is human molecules sticking to the surface. So remember, right at the beginning, I said, bacteria like to make biofilms on surfaces because nutrients concentrate on surfaces, right? Things just tend to stick to surfaces. So what often happens is you'll have human molecules deposited first, and then bacteria are attracted to those human molecules because bacteria that live on us and in us have specific receptors for human molecules. So like I said, if you make a surface that's eluting nitric oxide or antibiotics, you can reduce the load of the bacteria because you kill them as soon as they try to attach. But you are also then risking exposing them to sub-inhibitory concentrations, which can lead to resistance.
Do you know of any incompatibilities with different materials like silicone, polyurethanes, and so on?
That's a good question. So people have modified all kinds of surfaces. Usually the first consideration is biocompatibility with the person, so it has to be human biocompatible first, and then they worry about whether you can graft other things onto that type of surface or modify the topology of that surface. So silicone and polyurethane are by far the most common. There are also metal surfaces though, right? So more in knee and hip replacement type situations, you have titanium. So I remember I mentioned earlier that having a biofilm on a totally implanted device like a hip or a knee is a disaster, right? Because you can take a catheter out, but it's hard to take a knee out. I mean, you can do it, but so what orthopedic surgeons often do is use antibiotic impregnated cement when they're installing those devices in the first place. And that's supposed to help mop up any bacteria that might accidentally contaminate the surgical site. But even then, sometimes you get contamination. But it's sometimes hard to tell the difference between that type of, if you have pain at a site like that, sometimes it's hard to tell if it's infection or just inflammation. And so, that's where diagnostics come in. So people are trying to develop diagnostics that can tell you, is that pain in my hip due to bacteria or is it just sterile inflammation, right? So that's an interesting subfield of this biofilm study. And one of the things that people have tried, for example, is technetium-99 labeled ciprofloxacin, which will accumulate at your hip if that's bacteria, because it will go into bacteria, but not if it's sterile inflammation.
Interesting.
All right.
Do biofilms grow inside and outside of catheters?
Yes. That was the easiest question I've had yet.
Oh gosh. The article you mentioned. We've had a couple of questions. Could you mention where that can be found again?
M-Sphere 2018. The first author is Liu, L-I-U.
Perfect.
I don't have the slide in front of me anymore because I stopped sharing, but the reference is on the slide. So if you take a look at the slides once Ava posts them, they can get the paper.
I think Blake's frozen. He looks very frozen here. I'm not. I'm not. Do you think leaving biofilm in a catheter is a good idea for immunocompromised patients? If a treatment or preventative solution available, should that not be used?
Yeah, so anybody who's immunocompromised, that's a tricky situation because, again, I think it would, I'm not a clinician, but I think it would depend on what the organism is that is contaminating the device, because if it's something relatively innocuous, then I would be less concerned than if it's Pseudomonas. So, if you have a Pseudomonas or an Acinetobacter on a device that's in an immunocompromised person, that's a bad situation.
Now, a couple other questions are coming in specifically about the coating on catheters, whether silver is better versus CHG is better. Do you have an opinion?
They both work well. But as I said, we are starting to see silver resistance. And so partly that's because so many people are using silver everything, right? I mean, your towel at the gym now has silver in it, for God's sakes. Your socks have silver in it, right? So we are overloading the environment. Humans have this horrible tendency to go crazy on stuff, and basically we wreck it because we are selecting for silver resistance. So, you have the consideration of, you have potential silver resistance on one hand, you have potential chlorhexidine sensitivity on the other hand. So, it's going to depend on where you're putting the device and in whom you are putting it.
You're unfrozen, Blake. Welcome back.
I know. I'm trying to figure out where we're at with the questions.
I've been jumping a little bit. It just.
That's right.
So I'll pick another one, and then you go grab the next one after that. How's that?
I want to say thank you for everybody for hanging on for an extra half an hour. That's awesome.
No, I know.
I could talk biofilms all night long.
with joy, no doubt. And we still have 300 people hanging out. It's amazing. So thank you, everybody. When we're looking at central line infections, there's there's a lot of controversy. Do we take that line out? Did we wait a couple days? Is there any information that you have on that? Is there putting a new catheter in the same day, wait for a couple days, kind of clear out a little bit of the bug?
Yeah, that's that again, very patient and situation dependent, right? I mean, how badly does that patient need that line?
True.
And do you have an alternative site, right? So if you have a bad infection at one site, can you put it elsewhere or no, right? If, I personally, I have been a cancer patient, right? So I am not unfamiliar with lines, and, I did not want to have more than one installed.
No doubt.
So I would rather they try to recover the existing line if it was me, but again, that's up to the physician, the patient, and the situation.
Is extraluminal contamination a greater threat to a peripheral IV or any kind of IV catheter for that matter than intraluminal?
Greater threat in terms of the likelihood of it happening, yes. A greater threat in terms of what it might do to the patient, probably a little bit lower than intraluminal, because, blood passing through the line is the shear force will break off biofilm that's inside and carry it elsewhere, right? So we see this a lot with artificial heart valves. That's one of the examples that I mentioned earlier, is you get vegetations on these valves and they will break off and cause an embolic shower, and nobody needs that. So in that case, they're kind of flapping around because of the heart, the blood flow from the heart. But at a peripheral site, I don't know if that would be as big of a problem.
If I had an uncapped IV, how quickly do you think the bacteria are going to run up that catheter?
Depends on the bacteria. Again, so the one I showed you that video of, that Pseudomonas is very good at crawling across surfaces, right? It's aggressive. Things like Staph aureus or MRSA are not really motile. So their spread is more, as the culture grows, it grows up the catheter. I love that zoom effect.
I do too. That is very cool.
Have you heard of the antibiotic combination of minocycline and rifampin? This technology is being used effectively on catheters for anti-infective purposes.
Yes, I've heard of it. Again, I don't love the idea of antibiotic-coated catheters because we are desperately trying to conserve antibiotics. And when we expose bacteria to them, we select for resistant bacteria. So I realize that there's a place for this kind of thing, but it's not my favorite approach.
Are there current combinations being tried? Surfaces plus EDTA plus antibiotics?
People have tried all kinds of combinations. You know, if you look, just Google antimicrobial catheters or antibacterial catheters, and you will see what's out there.
If we treat an infection on a person with a central line, would this action increase biofilm on lines of the patient? I guess they're looking at cultures before writing a prescription for antibiotic therapy.
Yeah, that's a good question. So we don't, you know, we haven't studied this problem of provoking antibiotic or provoking biofilm with sub-inhibitory antibiotics in a mammal yet. So the data that I presented are what we see in the lab. But I know, so the nearest example I can give of where this might be a problem would be in cystic fibrosis patients. So cystic fibrosis patients have bacteria growing in their lungs because they have a mutation that makes the mucus in their lungs really thick so that they can't clear bacteria the way we would. And they, in order to kill those bacteria, they have to actually inhale antibiotics because you can't get a high enough concentration into the lung through a venous route. And you can imagine that there was a lot of concern that when you inhale an antibiotic, you're going to have a very high concentration up here, but you're going to have a difficult time getting it into the lower lobes of the lung, in part because of all the junk that's in there, the mucus and the bacterial debris and blah, 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 right? So the concern was that you might actually make the biofilm in the lower parts of the lung worse by causing this drop in antibiotic concentrations. I know that was a roundabout way to not answer the question, but.
I love it. So we have a couple of attendees that say, have mentioned pediatric patients are using 4% EDTA every day in Canada. And we also have someone saying it has large safety margins. Now, please verify that with our sponsor again, but just because we've had many questions on it, I wanted to share with you. I will ask you a question though. Push pause technique of a saline flush, does that have an effect on biofilm?
That's a good question. I'd have to think about that. I could think that you might. So one of the things we know about biofilms, which I didn't mention, is that the more shear force you apply to a biofilm, the tighter the bacteria will stick to a surface. So if you develop a biofilm in a low shear environment and then jack the shear up, it will often break off. But if you do it the other way around, if you develop it in a high shear environment and reduce the flow, nothing will happen, right? So the bacteria they respond to the mechanical cues of flow, right? And in fact, Pseudomonas has been shown to actively go upstream against flow. So if you mix it with another bacterium that can't crawl on a surface the way it can, it will turn the corner and go upstream, because that's where the source of food is coming from usually, right? If you're flowing media across a biofilm.
This is tricky stuff.
Yeah.
Bacteria are so cool.
I can tell.
I know you guys are all about killing them on your lines, but I think they're cool.
I grew up for a living. What about cat flow? So this kind of falls in line with that. We talked about the shear environment, push pause. If we're using a medication to help bust up a clot, so a catheter's become occluded, it probably has biofilm on it or in it. What happens when we break up that clot using a medication like Alteplase or cath flow?
That's a good question.
Again, I clot in the catheter, or?
Yeah, I mean, if there's a clot in the catheter and there are any bacteria around, they're probably sticking to the clot. So you would sweep them away with the clot when it's broken. I don't know if those compounds themselves would do anything to the bacteria one way or the other, because those are usually enzymes. Yes.
Yeah, it's designed to break down the matrix of blood clots. dissolving it. And the question was really, would that push it upstream into the bloodstream? And I would say, like you said, if it's growing on the obstruction and you broke it up, it would probably do
Yes.
exactly what that is. So it would go into the bloodstream.
Yeah.
So, yeah.
Yeah, I mean, a lot of the sequelae that come from, breaking biofilm loose are going to be dependent on what the organism is and how much of it gets broken loose, right?
How much damage would that do downstream? And really, that's dependent on the bacteria and the amount of bacteria.
Mhm. Yeah, I mean, we with the artificial heart valves, I know that can lead to stroke if you have small enough pieces breaking off that get lodged in capillaries, right?
Yep, it's the whole emboli, right?
Mm.
Yeah. In using the enzyme, would this release the biofilm into the patient? So very similar if TPA,
Mm-hmm.
also alteplase used to declot. Does this release biofilm into the patient? Yeah, so very similar
So
question.
the idea with those, remember I showed you, like, you add the enzyme and the biofilm basically dissolves, right? And the idea is that you are releasing not giant clumps of bacteria, but individual bacteria, or you're freeing very small
Segment.
sized clumps, right? And the smaller the clumps, the more easily dealt with they are by the immune system, right? So one of the ways that bacteria resist the immune system in biofilms is just because as a mass, they are bigger than a neutrophil or a macrophage, and they cannot be taken up, right? So if you can get that bulk down, then you have your immune system to back you up.
What if we're plugging around the catheter? Does that help at all reduce biofilm formation? And really, I think the question is related to things like skin glue or tissue adhesive, something that you would use to kind of like coat that insertion site from allowing skin bacteria to continue to enter into when
Yeah,
you use
that's
tunnel effect.
an interesting question. I would, it depends on what those materials do in terms of the bacterial load at the site, I would say. I don't know that a lot of people consider bacteria when they're developing these agents, right? They're thinking of what it can do in terms of keeping the line stable or in place, but not necessarily which bacteria would like to grow on this material now that I've coated it on your skin.
So we're not in the CE portion of the program now, and a couple people have asked, what is 4% EDTA, the name that people goes, and it's kite lock.
Kite lock.
So just wanted to make sure
Like
that you
flying a
guys,
kite.
and locking it on the fence. There we go. If biofilm grows inside and outside of catheters, how can 4% EDTA application help get rid of all biofilm? And then why use it?
I think it's more useful for either preventing or eradicating biofilm in the lumen, right? Which is really the part you're most worried about. You may still get biofilm on the outside, but that's less of an issue because you're not flowing that anywhere.
Okay. Oh, these two questions are great. Sorry, Blake, I'm going to jump in because I want to hear the answer. If we strip a line, if we go up femorally and we come in with a loop and we strip the biofilm, what time frame will it return? Plus or minus? No. Stop.
I would say, how well does brushing your teeth strip your teeth of biofilm? You may debulk the biofilm, but you're leaving behind bacteria that are just going to grow back, just like
Right.
just like the plaque on your teeth does.
How much do you really remove at that microscopic level? Yeah.
Exactly. And we, when we try to um, actually count how many bacteria are on a surface in a biofilm, it's hard to do because in order to count them, you have to remove them. And it's very difficult to do that. So, we try to, that's why we use these indirect methods, right? So we have a sonicating water bath, and we have tried to sonicate bacteria off a surface and then dilute them and count them. But, we're never able to get every single cell off of the surface ever. So we don't even try anymore. The bacteria win. Uncle.
So
That health wins, right? Just bacteria.
based on
What,
what you're talking, oh, sorry, Blake, I'm going to
no,
jump you in here.
that's-
I'm super interested on this other aspect about overwire exchanges. Are you familiar with that, Lori?
Over wire exchanges.
So we have a catheter that's either malpositioned or for whatever reason it's too short, it may have gone up the wrong direction, or there's an issue with it. We can't restore the patency. So we pull a bit out, we put a wire through it, then we put another catheter over it. Based on the knowledge that we're learning about biofilm, what are your thoughts about that practice?
I think you have to be exceptionally careful
It sounds like it.
with your sterile technique.
Ah, okie dokie.
I think that kind of falls into another question here, which is, if biofilm forms in the subcutaneous tracts of some of the tunneled lines that we place on patients. So.
Yeah, that does happen. I'm most familiar with PD catheters, peritoneal dialysis catheters, because you have a cuff that keeps them in place in the abdominal wall, and those cuffs often get contaminated. But again, depending on what it is, what's growing in there and how much of it there is, it can be a very slow burn problem or a rapid, oh God, we need to get this thing out of here problem, right? So, the bacteria on your skin are not horribly pathogenic if you're a healthy person, but, we're talking about people that have other comorbidities, right? So they're often immune compromised for other reasons. And so even staph epi, which is on your skin, can be a problem in those patients because it's getting into the wrong site. And their immune system can't adequately deal with it.
There's any Aussies on our webinar today, type in if you know if EDTA is available in Australia, if you would, please. How concerned are you with the use or overuse of antibiotics during COVID-19 and antibiotic resistance?
Huge.
Massive?
It's huge, tremendous.
It's horrible. So, first of all, don't get me started now. First of all, this whole hydroxychloroquine azithromycin thing was a terrible idea, right? So that got wildly promoted with very poor scientific evidence for efficacy. So azithromycin is a fantastic drug that we need to keep using. So we shouldn't be using it in the wrong way. That's point one. The second point is, unfortunately, severely ill COVID patients, almost all of them are getting antibiotics because they're mostly being ventilated, right? So you're going to give them prophylactic antibiotics to prevent VAP, or they're getting VAP and they have to be treated. So, in the early studies that were coming out of China, I think in the 90% were getting antibiotics of severely ill patients. And I don't think that has changed a whole lot because most physicians would not be comfortable venting a patient without coverage, right? It's
True.
Your mouth and your respiratory tract you're full of bacteria, and that's just a highway into your lungs. So, unfortunately, it's going to be a problem. We're already struggling with antibiotic resistance, and pharma is divested itself of this particular area. So, there are some glimmers of hope because there are biotech companies still working on antibiotics, but I can tell you that the two that most recently had an FDA approval of a new antibiotic, both of them went bankrupt because they couldn't sell enough antibiotic to recoup their investment in their clinical trials. And that's because when a new antibiotic comes out now, hospital formularies will buy, a few doses, and then they'll just put them on the shelf and they'll say, okay, we're not going to use it unless we absolutely have to. Well, how are you supposed to sell enough to make your money back?
Wow.
The attitude, right? So, one of the things that we're trying to work on now is to encourage governments and policymakers to come up with a different way to reimburse drug makers for making antibiotics. So, we're trying to encourage them to think of an antibiotic like a fire extinguisher, right? You don't ever want to use it, but you need to have it just in case. So who pays for fire extinguishers?
I have 2 in my house.
And do you want to use them? Of course not. But
I don't.
but
No.
you have them just in case, yeah.
That
So
is fact.
the UK is actually trying what they call the Netflix subscription model. So they are asking hospitals to pay a subscription fee that will be used to reimburse the drug maker, and that will allow them to have access to the drug if they need it. So we'll see how that goes. But there's a lot of changes that need to take place in order to make sure we still have antibiotics in the long run. Because we can't do medicine without them.
No, not effectively. We have a few more questions. We're winding down. Concentration antibiotics you need to use to kill biofilm. Mm-hmm. Is this common knowledge amongst doctors and pharmacists?
That's a great question. I would say no. So early studies that were done comparing, a planktonic version of an organism and a biofilm version showed that sometimes you need 1,000 times the concentration of a specific antibiotic to kill the biofilm, like to basically kill all the cells in the biofilm. And, that is mind-boggling to me. So, I mentioned early that it's crazy that we use planktonic cells to determine the MIC when ultimately we're dealing with a biofilm infection. So, we're kind of shooting ourselves in the foot by testing antibiotic concentrations that will kill susceptible cells when we have to kill resistant cells. So that was actually one of the reasons that MBEC lid device was developed, was to try to make an MIC test that was more realistic, using biofilms and testing the MIC directly on the biofilms to get the MBEC. Sorry, that's a lot of acronyms there.
No, it was great.
I did define them.
Are we headed towards a post-antibiotic era? And what is the role of biofilm?
Wow. Yes. Yeah. Yes, we are, but hopefully we can forestall that. So I actually wrote an article about this, at the beginning of COVID. So that it came out in April, talking about the other pandemic, right? Because AMR, antimicrobial resistance, is a pandemic. It's just a lot slower moving pandemic than COVID, but it has all the same characteristics, right? It's global, it affects everybody. We need public health measures to slow it down. And, we're all in it together, because if people don't use antibiotics properly, we're going to lose them. And think of all the things that we use antibiotics for, like routine things, right? Premature babies, cancer treatment. What would you do if you didn't have an antibiotic under those circumstances?
Die.
We would go back to a pre-antibiotic era. Nobody wants to go there, right? No. So we are working very hard to preserve the drugs we have and to find new ones and to use alternate therapies, right? So phage, I mentioned. People are trying other strategies like antimicrobial peptides. So these are modeled after what your neutrophils use to kill bacteria, right? So you can extract those and add them to the bacteria.
Will EDTA be considered a device or a drug?
I may have to leave that one for sterile care to answer.
Yeah, I think they'll probably pop in here and tell me in a second. Yeah. If higher concentrations of antibiotics was given to patients to treat infections, would the duration of treatments be shorter than reducing the length of time CVADs are in place, then reducing the growth of biofilm?
That's an interesting question. So there's a group of infectious disease specialists that are promoting the idea that we should be treating patients for shorter durations with antibiotics, right? So, where did 7 days come from? Is that just is there scientific evidence that you need 7 days, and why not 8 days or 6 days or 3 days, right? So the research that I'm most familiar with has been with UTIs, urinary tract infections, and they're trying to demonstrate that shorter is better. 3 days is enough, and by treating for longer than 3 days, you don't have enough benefit to warrant the risk of creating resistant organisms.
Very good.
So I don't know how that would apply in terms of lines, because again, we're not, then we're talking about a medical device surface, which adds an additional layer of complication to this situation.
If one patient has a central line and is getting pressors, and another patient that has a line is for antibiotics, is it likely the patient not getting antibiotics will get biofilm formation sooner?
I don't know. That's a good question. I feel like I'm studying for my medical school exam.
I told you when we talked before that we're a bunch of nerds and have some really...
No, I love it. I love all these questions. I'm so happy that you guys have stuck around for so long. That's great.
Following up the CHG question about dressings, do you know about silver being used in dressings to kill bacteria?
Yes. So,
okay.
I think they're selling silver Band-Aids now. Honestly, I would go with a regular Band-Aid and a topical ointment rather than put more silver into the world. I feel about silver like I feel about microplastics, right? For the amount of assistance it gives us, it's probably a dangerous precedent.
When you say lock in line, is that with every time you use it or access the device? I guess what he's trying to ask is using a locking solution to replace normal saline.
I'm going to have to pass that one to sterile care.
Sterile care coming your way. And this is another product specific question, and really talking about 2 different modes. And I'm going to pass on that one on your behalf.
Thank you.
But on Ava's behalf, I want to thank you sincerely. This was a phenomenal talk. Love the Q&A, and we really look forward to having you back, because
I really enjoyed it. Thank you for having me.
We enjoyed it so much. So thank you. Everybody that stuck around, thank you so much. We have over 200 that stuck through all of this.
Great. Thank you, guys. And, stay safe out there. I know a lot of you guys are on the front lines, and we appreciate it.
We appreciate all the research you're doing. You're making us much smarter. So thank you.
We're doing our best.
Okay. Take care, everybody. Thanks for being here.